let's just finish the session, and then we go through the review slides, right? I just wanted to go over again the branched chain amino acid metabolism. So three amino acids, leucine, leucine, valine, are branched chain amino acids, right? Did you guys get the revised slide, right? I revised actually many slides. The branched amino acids metabolism. This slide, I think I did something wrong last time, so just wanted to remind you again. Most of the branched chain amino acids not metabolized by intestine and liver, right? They bypass intestine and liver, and the majority of branched chain amino acids actually are metabolized in which tissue? Muscle, right? So what is the reason why the branched chain, the liver cannot metabolize most of branched chain amino acids? Because of the enzyme, right? The branched chain amino acids transferase, BCAAT, its activity is low in the liver. So the most of the branched chain amino acids do not get metabolized in the liver because of lower activity of BACCT. Okay, I can't remember what I did wrong last time. Maybe I said it a different enzyme, right? So try to remember it's BCAAT. Yeah.
Okay, so the last one you had written BCKAD.
Oh, that was wrong. Sorry.
Okay.
I apologize
Okay.
for that. So again, the BCAAT is an enzyme, right, that has a lower level in the liver. So that's the reason why liver cannot metabolize most of the branched chain amino acids, right? So after meal, the branched chain amino acids levels goes up significantly, right? You have higher levels of branched chain amino acid after meal, and excessive branched amino acids that beyond the need for protein synthesis, right, will be catabolized in the muscle particularly, right? So most of the branched chain amino acids are metabolized in the liver. So in the muscle. So muscle is the main site for metabolism of branched chain amino acids, right? Is that clear? Okay. And also modify this slide. This sort of kind of overall picture of the branched amino acid metabolism in the muscle, right? In the muscle. So like other amino acids, type of amino acids that you have nitrogen and carbon metabolism, right? The two different pathways. So nitrogen, first of all, undergo the transamination by BCAAT, right? We talked about it a little bit last time, right? We're going to go over again. So don't worry, I will send you this slide. That will be included in the review slide. And so nitrogen metabolized after remove the nitrogen, right? Then will get will convert to the glutamine. So glutamine and then undergo the further transamination by a enzyme, the alanine amino transferase, right? So ALT. This is also the transamination reaction. So then glutamine will convert to alanine, and glutamine can also be metabolized to glutamine, right, by a glutamine synthetase, right? So this sort of overall, pathway of nitrogen metabolism of branched chain amino acids. So branched chain amino acids actually undergo the two transamination reactions. First one is catalyzed by BCAAT, and the second transamination reaction catalyzed by ALT, right? But glutamine will also metabolize to glutamine by glutamine synthetase, right? And for the carbon metabolism, most important pathway or enzyme for carbon metabolism of branched chain amino acids is BCKAD, right? So BCKAD is the enzyme that catalyzes the oxidative decarboxylation, right, to further break down or further metabolize the alpha ketone acid. So alpha ketone acid will be catabolized further to either acetyl-CoA or propionyl-CoA, right? So also something that you need to remember that the BCKAT catalyzed reaction is type of reaction that called oxidative decarboxylation reaction, right? And the this two products of a carbon metabolism, acetyl-CoA and propionyl-CoA. Acetyl-CoA is ketogenic only, right? Cannot be used for gluconeogenesis. So acetyl-CoA is a ketogenic only, and propionyl-CoA, if you still remember, is a precursor of a gluconeogenesis, right? One of the precursors. So propionyl-CoA can do both glucogenesis and ketogenesis. So propionyl-CoA is glucogenic and also ketogenic, right? Does that make sense? And this is they'll put all pathway together of the branched chain amino acid metabolism. So the first reaction is a transamination reaction, right? Catalyzed by BCAAT, which is branched chain amino acids transferase, right? BCAAT. So BCAAT reaction, the first step is a reversible reaction. So reversible reaction means that the reaction can go the two directions, right? And the product for the first step is alpha keto acid, and glutamate is generated, right? So and for three branched chain amino acids can all be metabolized to alpha keto acid through the irreversible transamination reaction by the enzyme BCAAT, right? So BCAAT catalyzes the transamination reaction, right? And this reaction again is reversible. And step 2 is the again the catalyzed by BCKAD branched chain keto acid dehydrogenase. And this reaction, this step, this enzyme catalyzes I just mentioned, right, last slide, is oxidative decarboxylation reaction. And the final product of the BCKAD catalyzed reaction, acetyl-CoA, propionyl-CoA, or acetoacetate, right? Depend on which branched chain amino acids. So leucine only produces acetyl-CoA. So that means leucine is a unique ketogenic amino acids, right? Does that make sense? So leucine cannot be glucogenic. But isoleucine and valine can produce both acetyl-CoA and prominyl-CoA. So these two branch chain amino acids are ketogenic and also glucogenic, right? So and then I divided two slides, right, which was originally one slide. Just wanted to point out the key important points, right, for step 1 and step 2. So for step 1, we have a key point here. So what type of a reaction is it? For step 1, catalyzed by BCAAT. Transamination, right? Okay. So compare BCAAT activity in muscle and liver. Which tissue has higher BCAAT activities? Muscle, right? And liver is lower. So that's the reason why liver cannot metabolize most of branched chain amino acids, but muscle does, because muscle has a higher BCAAT activity. Okay, so what happens to the alpha keto acid of branched amino acids in the fat state? After the first step, right, the transamination reaction, the alpha keto acid is generated. So what is the fate, metabolic fate of alpha keto acid in the muscle under the fat fasting condition? So in the fat state, the alpha keto acid, the branched chain amino acid alpha keto acid, right? So you need to add the branch chain. So the transfer to other tissues for protein synthesis. Under the fat condition, branch chain amino acid alpha keto acid, once it's generated by BCAAT, will be transferred to other tissue for protein synthesis in the fat condition, right? And if transferred to liver, they actually are further metabolized for energy. So depend on which tissue, right? So in the liver, because if you still remember, the liver actually use alpha keto acid for energy, right? So the main energy source for liver is alpha keto acid. So even under the fat condition, once the alpha keto acid are generated in the muscle and transferred to liver, they are actually oxidized for energy in the liver. But if it transfers to other tissue, that will be used for protein synthesis, right? So during fasting, the alpha keto acid generated in the muscle will not be transferred to anywhere, but stay in the muscle for being oxidized for energy, right? Does that make sense? Okay. And cellular location of step 1 is actually in the cytosol, and both cytosol and mitochondria. Any questions? Is that clear? Okay. And step 2, the BCKD is the enzyme, right, to catalyze oxidative decarboxylation reaction and for generating acetyl-CoA, prominyl-CoA, or acetoacetate, right? Depend on, again, which branch chain amino acid. So what type of reaction is it? Oxidative decarboxylation, right? Okay. So compare BCKAD activity in muscle and liver in the fat and fasted state. The activity in muscle is also different. Remember in the last slide that we mentioned that in the fat state, most of alpha keto acid generated in the muscle are transferred, right, to the other tissue for protein synthesis. So that means alpha keto acid in the fat condition will not be further metabolized in the muscle, right? What does that mean to the BCKD activity? under fat condition, BCKD activity is low, right? So that means that cannot further metabolize alpha keto acid. Instead, alpha keto acid be transferred out of to other tissue. But it's much higher in the liver. So that's why once the alpha keto acid being transferred to liver and gets metabolized, right, by BCKD, right? Does that make sense? So try to connect all this together. Remember the enzyme activity in each tissue, higher, lower, right? And in the starvation or metabolic stress, the BCKD activity is actually higher in muscle than liver, because under starvation condition, the muscle utilize, right, alpha keto acid for energy. So the BCKD activity goes up, to increase, so then can oxidize alpha keto acid for energy in the muscle. So like almost 5-fold increase in the starvation or under the stress condition, right? And this reaction actually occurs in the mitochondria. Any questions about the step 2? Is that clear? So try to remember the activities of the BCKD, right, under fasting condition, compare the liver muscle, right, and connect with the alpha keto acid transported or oxidized, right? Okay, so now I have 2 slides, actually borrowed from Dr. Carrie Earthman. I think so. Pretty much, I just wanted to describe the process that I already described in a couple of previous slides, right? So again, this slide showed the BCAA, branch chain amino acid metabolism in muscle under the fat state, right? So first of all, so this just represents the muscle, right? Something happening inside of muscle cells. So first of all, the all 3 branch chain amino acids get the metabolized or gets transamination, right? Undergoes transamination by BCAAT enzymes, and generating BCAA alpha keto acid, right? And also glutamate. So glutamate will undergo further metabolized, the second transamination reaction, right, by the enzyme, the alanine aminotransferase, right? So transfer the amino group from glutamate to pyruvate. So the glutamate itself gets converted to alpha ketoglutarate, right? And pyruvate and converted to, once it's received the amino group from glutamate and converts to alanine, right? So so in this metabolic process, the alpha keto acid and alanine are generated, right, in the muscle. And glutamate can also be metabolized through a different pathway, right? Transamination is one pathway, and the glutamate can also be converted to glutamine by glutamine synthetase. So glutamine synthetase is also have a high activity in the muscle, right? And so carry the one amino ammonia. This ammonia basically is removed from other amino acid metabolism, right, in the muscle. So the glutamine is generated. So glutamine is the transporter of amino group, right? The nitrogen. So yeah, question. right?
That may change things that would make it, like, is that one of the things that was like originally wrong?
Which reaction you mean?
So
The glutamate?
yeah. So right there's ammonia.
Right?
On your original slide, it's ammonia. So is that a correction?
Right. So that will convert it back to the glutamate, right? Glutamine.
Okay.
Does that make sense? Yeah. Question. Oh, okay. Yeah. So depend on the pH, in the environment. So sometimes the H3, they can interconvert it with both form, the ammonium or ammonium, right? So okay. So the glutamine is also be produced, right, in the muscle under the fat condition. So what you need to remember, that's the three compounds, right? Alpha keto acid, alanine, and glutamine are made during the fat condition, right? From the branched chain amino acid metabolism and released into the circulation. So three compounds are made and released into the circulation. So the alanine or glutamine will be transported to other tissues, including liver and GI tract, like intestine and kidney, right? So the glutamine particularly usually being transported to intestine and kidney. Remember, glutamine is the most important energy source, right, for the intestine, and 40% of glutamine in the intestine from diet, and another 40% actually from the muscle, right? The glutamine in the circulation from muscle. And alpha keto acid and can be also released into circulation and transported to other tissue and get reamination for protein synthesis, right? But if alpha keto acid are transported to liver, because of the liver has a higher BCKAD activity in the fat condition, right? So it gets oxidized. Alpha keto acid gets oxidized in the liver because of the high levels of the BCKAD in the fat condition, right? Any question? Okay, now how about under the fasting condition? So similarly, this will also happening, BCAAT or ALT transamination also happening in the muscle under the fasting condition. Also the glutamine synthesis reaction or processes also occur in the fasted condition, right? So both glutamine and alanine will also release into the circulation, but the alpha keto acid will not be released into the circulation because of the BCKAD enzyme activity goes up or get increased under the fasting condition, right? So alpha keto acid will get further oxidized in the muscle, will not transfer out of the muscle, right? Again, this is a product depend on which branched chain amino acids. And if you I don't know if you notice that there are actually only two branched amino acids listed here, valine and isoleucine. We have one more leucine, right? So what happened to the leucine during the fasting condition? So leucine will actually get completely oxidized in the muscle to CO2, water, and produce ATP, right? Because the final product of losing metabolism is acetyl-CoA, right? So acetyl-CoA will get to the TCA cycles and completely oxidized. All right, does that make sense? So under the fasting condition, muscle only releases the two alanine compound, alanine and glutamine, right? And not alpha-keto acid. And because of, again, BCKD activities increase, so alpha-keto acid gets oxidized immediately in the muscle. Okay, so clinical relevance, the maple syrup urine disease. Have you ever heard about this disease? So basically, the urine smell kind of sweety smell, smell like maple syrup. So the cause of this disease is a deficiency in the BCKAD, right? So BCKAD is lacking in the patient. So you know what BCKAD does. It's to metabolize branched chain amino acids, right? So what will happen if the patient lacks the BCKAD enzymes? A patient be able to metabolize branched chain amino acids? No, right? So you have lots of branched chain amino acids, alpha-keto acid accumulated. So that is the reason causing the disease. Right. So summary: the BCAAT is a key enzyme for transamination reaction of the branched chain amino acids in the first stage of metabolism, and BCKAD is a key enzyme complex for branched chain amino acid oxidation in the second stage, right, metabolism. And both enzymes have a different activity in the liver and the muscle, under the fat fasting condition, right? So it's a little bit complicated. You probably want to remember, right, the activity of each enzyme in liver and muscle under the fat fasting condition. And glutamine and alanine are the primary product of the BCAA metabolism in muscle, right, and be actually transported to other tissue for further metabolism, for protein synthesis or oxidation in the liver. And BCAA alpha-keto acid have a different phase, right, metabolic phase, depending on physiological condition, right. Under the fat condition, alpha-keto acid will be transported to the other tissue for protein synthesis or liver for oxidation, for energy. Under the fasting condition, the alpha-keto acid will immediately metabolize in the muscle, right? Okay, so we're also going to talk about some specific amino acid metabolism, glutamine, glutamate, and aspartate. So glutamate and aspartate play a central role, right, in the nitrogen metabolism. So glutamate serve as sort of important the nitrogen transfer, right? So nitrogen and metabolism usually through the two important type of reactions, transamination and deamination, right? And these are the enzyme, the aminotransferase and the glutamate dehydrogenase. These two are the enzyme, right? The transamination for the aminotransferase is for transamination reaction, and glutamate dehydrogenase it catalyzes the deamination of glutamate, right? So most the glutamate and aspartate metabolism is intracellular. So the metabolized in the inside of cells, right? The involved in the transamination and deamination. And glutamine is catabolized predominantly by deamination, right? Glutaminase is an enzyme to catalyze the deamination of glutamine. So glutamine metabolism is both intracellular and in the circulation, right? Glutamine is usually present in the circulation by very high levels, right? And glutamine, I also mentioned, is one of the amino acids that serve as the nitrogen transporter, right? Okay, so glutamine metabolism in the muscle. So glutamine is converted to glutamine is actually converted from the branched chain amino acids, right, and released into the circulation, just like we described in the muscle, right? The muscle produce a lot of glutamine from branched chain amino acids metabolism. And so the release of a glutamine from the muscle usually serves to transport branched chain amino acids, carbon and nitrogen, right, to intestine, immune cell, kidney, and liver. So muscle has a high levels of glutamine synthetase, right? So this sort of pathways for the glutamine generation, right, and release into the circulation and goes to these tissues for either use energy or for the further metabolize. Right, so start from the valine and isoleucine, the branched chain amino acids being metabolized to succinyl-CoA and oxaloacetate. So this is part of a TCA cycles, right? The citrate and then the alpha-ketoglutarate. So alpha-ketoglutarate then be converts to glutamate through the transamination reaction, right? And then glutamine synthetase and converts the glutamate to glutamine. So this is how a glutamine is synthesized from branched chain amino acids, right? This is a pathway. And glutamine can also be metabolized in the intestine. If you still remember, glutamine is the most important energy source, right? So glutamine, first of all, it gets metabolized by deamination, right? The glutaminase and remove the ammonium, right? And converts to glutamate, and then glutamate undergoes the transamination by amino transferase and gets converted to alpha-ketoglutarate. So alpha-ketoglutarate is part of is a TCA cycle intermediate, right? So can enter the TCA cycle for complete oxidation for ATP production, right? So this is the pathway for glutamine utilization for energy, right, in the intestine. Okay, so this is last slide. The interorgan glutamine metabolism. So you probably wanted to know which tissues produce glutamine and which tissues utilize glutamine, right? So for example, most of the glutamine actually produced by muscle and also liver and brain, right? So muscle, liver, brain are the contributors of a glutamine, right? They make glutamine and release into circulation. And the gut or the intestine, again, is a major users of glutamine, right, for energy. And also kidney, lots of glutamine will also goes to the kidney for releasing the ammonia, right, in the kidney, so get rid of ammonia. And also the immune cell. Immune cell also utilize glutamine for energy, right? So you probably wanted to understand which tissue produces glutamine and which tissue utilize, right? Okay, that's all I think for the amino acid metabolism. Now let's go to the review session. Any questions? So so something that you probably wanted to know for the exam, amino acids, right? The essential amino acids, non-essential, and semi-essential. So semi-essential basically is amino acids that can be synthesized from non-essential in the body, right? And we also, I realized that we also have, okay. So in the study guide, in question number 9, we're talking about the conditional essential amino acids, right? So what are the conditional essential amino acids? The amino acids usually is not essential, right? But become essential, under the certain conditions of illness or stress, right? So arginine, glutamine, and cysteine are the examples of a conditional essential amino acids, right? For example, the arginine, when the arginine is essential for the patient suffers from cancer, right? So cancer patient usually requires more arginine. So your body are able to synthesize some arginine, but cannot meet that requirement under the cancer disease condition, right? So that you have to provide arginine from the diet, more arginine, right, from the diet. So under the disease condition, the arginine then become essential, right? So that is the conditional essential amino acids. I also have a collection for question number 15. Sorry, I messed this up. So it's actually the alanine transferase, right? ATL is alanine transferase, and AST is aspartate aminotransferase, right? I will post it on this slide. And in general, the amino acid metabolism, right, under the two different pathways, the nitrogen metabolism and carbon metabolism. So you wanted to understand. So the nitrogen metabolism, the two important reactions, right, the catalyzed nitrogen reaction, they are transamination and deamination, right? And you also wanted to know what the purpose of the transamination and what is it. We have two examples, right? ALT and AST catalyze the alanine aspartate transamination reaction, right? So you wanted to remember the substrate and a product, in each side. And nitrogen, once it's removed from amino acids, that need to be excreted, right? And by the process called urine cycles, right? For synthesize the urine and get rid of the urine through the urine. But kidney can also directly excrete the ammonium, right? So it's also the way of the getting rid of the ammonium. And the brain can also incorporate ammonia into the glutamate or glutamine, right? So several ways, but urea synthesis is the most important pathway for excretion of nitrogen, right? And carbon skeleton, it can be also metabolized, serve as for energy use, right? In the different tissues. Okay, so transamination reaction. This is a general reaction. So you also want to know that glutamate alpha keto acid, right, is the PLP, right? Basically, the vitamin B is important for the transamination reaction enzyme activities that involve this. And also, I wanted to remind you that this transamination reaction is as reversible, right? So it can go two directions. So you wanted to remember the two transamination, like, if you want to go this direction, what is the product, and a substrate and product, right? And if you go to the other direction and the product, what are the product and what are the substrate and product, right? So, and you also wanted to know that alpha ketoglutarate and glutamine played a very important role, right? It's the most common the acceptors of a ammonium group, right? Alpha ketoglutarate. And it's just a different transamination reaction. And key point about the transamination, so why it is important? And because this is a way to remove the nitrogen, right? Or ammonia. And to allow the carbon skeleton be further metabolized, right? And the transamination, these are the two enzyme transamination reaction enzymes that are very active, right? ALT and AST, right? Alanine aminotransferase, aspartate aminotransferase. These are the two most active transamination enzymes, right? And the key amino acids, the intracellular hub for transamination reaction is a glutamate, right? And here I wanted to remind you, I actually revised this, sorry. So the reversible, again, the transamination is a reversible reaction, right? So which direction predominates in the fat state? So in the fat state, the transamination basically moves in the direction of formation of glutamate from right to left. From left to right, right? So and moves towards in the fat state, right? Move forward in the fat state. And which vitamins require vitamin B and so this is also important point that not all the amino acids undergo transamination at the first step, right? Some of the amino acids just directly go to deamination, right? Which amino acids did you guys remember? Threonine, right? Let's go directly to deamination. Does not, undergo the transamination. Okay, so this is a deamination. The enzyme for example, threonine can directly undergo deamination by threonine dehydratase, right? This is so you remove the one ammonium and threonine converts to alpha ketoglutarate. Okay, so glutamate dehydrogenase is very important enzyme for deamination, right? Because lots of alpha ketoglutarate will use the common useful acceptor of nitrogen, right? Remember, so through the transamination reaction of many amino acids, the glutamate will generate, right? A lot of glutamate is generated. So glutamate undergoes deamination. So by this enzyme, glutamate dehydrogenase. So that's why the glutamate dehydrogenase is so important, right? Because you have lots of glutamate is generated from transamination reaction, right? So the alpha ketoglutarate is a product. Yes.
I had a quick question from the study guide. It says which two amino acids do not undergo transamination at the first step of the metabolism. We just talked about three.
Yeah, they also, if you go back to that stuff, sorry. Yeah.
Multiple.
Hmm?
So there's multiple.
Right,
So
right.
you don't need to know a specific two, because here it just says which two.
Yeah, you can pick of two of them. Screening, yeah.
Okay.
Yeah, okay. Okay, so deamination, right? And glutamine also produce, right? Glutamine has a two amino group, if you still remember. So under also need to be, the ammonia amino group also need to be removed from the glutamine. So glutamine again is one of the nitrogen transporter, right? It's a very important nitrogen transporter, and you generate from the tissue, particularly muscle, liver, and brain, right, and release into circulation and pick up by other tissue, for example, by kidney. So the purpose of the glutamine is transport nitrogen or the ammonia from one tissue to the other tissue and get rid of the ammonia, right? So once the glutamine is arrived in the kidney, then glutamine undergo the deamination by glutaminase and remove the amino group and release amino group. So if in the kidney, that this will be converted to ammonium, right? The NH4, and then excreted in the urine. But if if this reaction occurs in the liver, then the ammonium will undergo urine cycle, right? Synthesize urine. Okay, and it's just the overall flow of the nitrogen metabolism, right? In the so first of all, undergo the transamination, and second step is the deamination, right, by glutamate dehydrogenase, and then remove the removed ammonium that enter the urine cycles and for synthesis of urine, right? So this is to undergo transamination and deamination and the urine synthesis. So what is the fate of ammonium that generated from the nitrogen metabolism, right? You just mentioned. Primarily go to the urine cycle for synthesis of urine. So urine cycle is the most important mechanism, right, to get rid of ammonium. And also glutamine synthesis. Glutamine is very important, right? The carrier of the nitrogen or ammonium, right, from one tissue to the other tissue, particularly to the kidney, and for the release of the ammonium. Yeah.
Which tissue does glutamate synthesis
That's a good question. So let's see. I think that we have actually a slide in the last. I forgot to mention. Let me go to that slide. Okay, so so try to remember this slide, right? So glutamine synthetase, right? What's that? Right. Liver muscle is pretty, and the brain, right? So they have a higher activity of glutamine synthetase. Does that make sense? Because these are the three main tissues that produce the glutamine, right? So you will expect the glutamine synthetase. Well, this enzyme probably not that regulated by like fasting state, because in both conditions you will generate a lot of ammonium, right? So it's not so much regulated by fat fasting, right? Okay, let's go back that slide. Okay, so the two ways, right? The first mechanism, the two mechanisms, right, to get rid of ammonium. One is the urine cycle, most important pathway or mechanism, and the glutamine synthesis, right? So glutamine synthetase is the enzyme that catalyzes the synthesis of glutamine from glutamate. So you're adding the ammonium to the glutamate to make a glutamine, right? So this is also the way to carry, the ammonium from one tissue to the other tissue. So remember, this reaction is irreversible, right? Glutamine synthetase reaction is irreversible. You only go to one direction. For synthesis of glutamine, you cannot go back, right? And also, this is not the transamination or deamination reaction, right? So glutamine synthesis reaction is not the transamination or deamination. Although you're adding the ammonium to the molecule, to that glutamine. So two questions here. Amylo group is removed and ammonium is produced. How do we get rid of ammonium? Urine cycle, right? And glutamine synthesis, right? So what happened to the carbon skeleton? Right? Carbon skeleton will get further metabolized, right? Oxidized. Okay, so urine cycles, and you wanted to know the two steps in the mitochondria and 3 steps in the cytosol, right? And the substrate, the first step, substrate is 1 ammonium, right? And bicarbonate or CO2, right? Feed into the reaction. And first step catalyzed by enzyme CPS1 and several cofactors that are important for CPS1 activity, right? NAC is one of them activities. And the second step is the OCT, right? The enzyme catalyzes the generation of citrulline. So citrulline is generated from the first two steps of a urine cycle in the mitochondria and released into the cytosol and continues the reaction, right? The rest of the three steps in the cytosol. And the second second nitrogen in the urine is coming from aspartate, right? And you probably wanted to remember the enzyme, that for the nitrogen, for the feeding nitrogen into the cycle. And lastly, the arginase, right? Arginine is break down the, arginase break down arginine and to release urine and the ornithine. So ornithine gets recycled back to the urine cycle, right? So liver can do complete urine synthesis, urine cycles, and the other two tissues only does the partial urine cycles, right? What are the two other tissues? Kidney and intestine, right? Okay. And also you wanted to remember the urine cycle, which type of cells in the liver that does the urine cycles. Periportal hepatocyte, right? Okay, so the this I think that's the carbon chain, metabolism, right? Depend on the fat and fasting state, how carbon skeleton of amino acids being metabolized. So there are several ways, right? Several pathways. One is for synthesis of glucose and glycogen, right? And also synthesis fatty acids and ketone bodies, right? And depend on, again, the fat fasting condition. So you wanted to remember this diagram or slide, very important. Try to understand how, fat fasting condition, how carbon is metabolized for, again, not only depend on the energy state, but also depend on the abundance of amino acids, right, in the body. So then can go, carbon skeleton can go to the different metabolic pathway, right? So I'm not going to repeat this. And also want to mention that gluconeogenesis is also occurred in both in the fat state, right, for the amino acids metabolism, and the glucose that's synthesized from gluconeogenesis are incorporated into the glycogen, right, under the fat condition. Okay, that's basically the similar thing. So, and for the ketogenic amino acids, I know you guys know the leucine, right? And lysine is also the another one. But if you can remember, that's okay. If not, it's fine, right? So, all right. Yeah, I think that you can probably want to just go through all these slides, in terms of energy utilization in the intestine, if you still remember, 80% of the energy, right, from the glutamine, and aspartate, right, for the intestine. This is just the metabolic flow of amino acids in different tissues under fat fasting condition. So you see that under the fat condition, the 20% of the branched chain amino acids will go to the liver, right? But liver can metabolize some, but majority of branched chain amino acids get metabolized in the muscle, right? And glutamine is released from glutamine alanine are released from muscle, right, from the branched chain amino acids metabolism. And glutamine basically goes to actually majority of glutamine actually go to the kidney and the intestine, right, and not much to the liver. But alanine basically, most of alanine actually go to the liver. So different amino acids, they have a different metabolic flow in different tissues, right? And fasting condition, that also, you produce a lot of alanine from kidney and particularly muscle, and also intestine, that all go to the liver for gluconeogenesis, right? But glutamine will still go to the kidney and intestine. And also depend on the state of starvation, how the amino acids be metabolized. Right? So the early stage of a starvation, we usually use amino acids for gluconeogenesis, right, to raise the blood glucose level. But at the later stage of starvation, because we already break down a lot of proteins, right? We don't want to waste more protein. So we actually, at a later stage starvation, gluconeogenesis activity is, let's see, more accurate. So gluconeogenesis is from amino acids, this pathway being reduced. So we actually try to spare proteins, right, amino acids, in the later stage of starvation. Okay, so I think this slide is also important, so give you the overall idea how dietary amino acids get metabolized in the different tissues. For example, the first stop in the liver, right? So 60% of the dietary amino acids, here I'm talking about any type of amino acid, not just the branched chain amino acids, right? So 60% of overall dietary amino acids will get metabolized in the liver, right? And 20% of dietary amino acids were used for protein synthesis, and another 20% of amino acids will not get metabolized in the liver, released into the circulation and go to other tissue for metabolism. Well, all right. So again, a similar, thing. Yeah, I guess it's pretty simple slide. Yeah, it's just the overall, the connect all the pathway together, right? The amino acids gets the transamination by aminotransferase and a deamination by glutamate dehydrogenase, right? And to release ammonium, and ammonium then enter the urine cycles, right? This is just the details, right? So you probably wanted to know again the substrate, the first step, and the enzymes, CPS1, and the cofactors, right? And how many ATPs are generated? 2 ATPs produced from the first reaction, and the second reaction is the OCT catalyzed reaction, right? For generating citrulline. And this is all the important point. And the periportal hepatocyte, right, is a place where urine synthesis occurs, and it's first 2 steps in the mitochondria. And cytosol, you have the rest of the steps, important steps that feeding the nitrogen right into the cycles from the aspartate and by this enzyme, right, arginine succinate, what do you call it? Synthesis, right? And this step 3 also utilize 2 ATPs. So you have 4 ATPs already being utilized, and most important thing is that the fumarate, right? Fumarate, it gets also recycled, right? So you wanted to understand what intermediates, right? Compounds that involve in the fumarate, for example, malate, oxaloacetate, and aspartate, right? So aspartate, go back to the cycles. So all these components, right, are involved in the fumarate. But most important thing is that fumarate cycles actually produce the 3 ATPs, so that the net utilization of ATPs is 1, right? During the urine cycles. The step 1 utilize 2, and step 3 utilize another 2. So 4 ATPs are utilized, but 3 ATPs are generated during the fumarate cycle. So the net utilization of ATPs for the urine cycles is 1, right? 1 ATPs is needed for urine cycles. Okay, so intracellular glutamine cycle, this is very important, right? So there are 2 types of cells that are involved in these cycles. One is the periportal hepatocyte, right? And the other one is the perivenous hepatocyte. So periportal hepatocyte is a main cell that does urine cycles, right? So urine cycles only occur in the periportal hepatocyte, right? Because of enzyme, very active enzymes that involve urine cycles are present in the peripodocyte, but not in the perivenous hepatocyte, right? And the perivenous hepatocyte basically converts the feed actually nitrogen to the glutamate and forming glutamine. So glutamine synthetase activity is higher in the perivenous hepatocyte, right? So this actually is the backup mechanism for removing ammonium, right? So again, the most of the ammonium molecules are removed by periportal hepatocyte through the urine cycles, urine synthesis, right? And just some ammonia that escapes hepatocyte that will be picked up by the perivenous hepatocyte, right? And then converts or incorporate into glutamine to form glutamine and then release back to the circulation, right? So this can be picked up by the kidney, right? To further metabolize and remove the ammonium and excrete it ammonium in the kidney, right? So again, the purpose of a grooming cycles is to remove the ammonium, right? And that involves two types of cells. So the paravenous hepatocytes serve as sort of backup mechanism, right? The main process of remove ammonium is in the periportal hepatocyte, right, by urine cycles. Does that make sense? Right? So I have listed several points to help you understand better of the grooming cycles, right? First of all, the periportal hepatocyte use or pick up ammonium to make urine, right? And second, paravenous hepatocyte use or pick up ammonium to make grooming, right? Different mechanism. And third, the periportal hepatocyte have higher glutaminase activities. Right? That is very important for release the ammonia, for the urine cycle, right? To feed the nitrogen to urine cycle. And next, the paravenous hepatocyte have a higher grooming synthetase activity, right? So can make a grooming. And periportal hepatocyte have a higher activity of urine cycle enzymes, right? And paravenous hepatocyte have a no active urine cycle enzyme. So paravenous hepatocytes are not able to do urine cycles. The way that the paravenous hepatocyte to remove ammonia is to produce or generate grooming, right? Does that make sense? So two different mechanisms, they work together, right, to try to get rid of ammonia as much as possible. Okay, and it's the regulation of urine cycles. The CPS1 activities, right, that regulate by ammonium concentration, the higher ammonium concentration, to activate CPS1, which makes sense, right, to increase the urine synthesis. And NAC is necessary, right, for CPS1 activities, right? And also the second enzyme in the mitochondria is OTC. So OTC activity regulates by product and substrate. So adenosine is the substrate, right? The OCT substrate. So adenosine levels increase, then stimulate OCT, and citrulline, which is a product of OCT, if its concentrations increase, then inhibits OCT activity, right? And urine cycle enzymes and hepatic glutaminase activity also regulated by the protein intake, right? So high protein intake increases activity of urine cycle enzymes, right? And also hepatic glutaminase, which makes sense, right? We get more protein than we need to metabolize the protein, right? To get, you produce more ammonium. So we needed to get rid of ammonium. So increase the urine cycle enzyme activity. Okay. Yeah, this basically is a question that you guys, I guess, all know, right? So the urine cycle, as urine synthesis, so where does the carbon comes from in the urine molecules? From bicarbonate, right? And the two nitrogen in the urine molecules are from ammonium and aspartate, right? Okay. So how many ATPs are needed for urine cycles? 1, okay. And so adenosine and fumarate gets recycled. Why is it important? Right, to get urine cycles running, right, functioning. So what regulates the urine cycle that we just talked about, right? Several factors: ammonium concentration, NAC, ornithine, and citrulline, and the high protein, right? It can all regulate the urine cycle activity. Okay, so clinical implication, basically, I think it's right. We have to get rid of ammonia, otherwise it's toxic to the brain. So if liver failure, that will causing the coma, right? The hepatic coma or something like this. So, yeah, that's just talking about the carbon skeleton metabolism, right? In the liver, basically, it's for gluconeogenesis, release glucose during fasting, or for synthesis of glycogen during fat state. And during the fat state, the carbon skeleton of amino acid can also be used for synthesis of the amino acids, the fatty acids, right? And during fasting, the carbon skeleton of amino acid can also be used for synthesis of ketone bodies, right? That is just a summary. So, metabolism in intestine. So intestine, we know that the glutamine is the most important amino acids, the energy source for intestine, right? Glutamine, aspartate, the three amino acids are the energy source of for intestine. And also some of the amino acids being used to synthesize protein, right? Like apoprotein and the other enzymes, digest enzymes that also needed to be synthesized from amino acids. And partial the urine cycle, right, occur in the intestine. So the glutamine, we talk about the 40% of glutamine in the intestine from diet, right, and another 40% from circulation. So which is the tissue that contribute the most of glutamine to the circulation? You guys remember which tissue? Circulating glutamine. The major contributors of the circulating glutamine. Muscle, yes, muscle. Right. Okay. So this is the amino acid metabolism in the intestine, right? So you wanted to know, which compounds that are generated from glutamine metabolism in the intestine and released into the circulation. Basically, as the ammonium, right? Of course, we removed from the glutamine, and ornithine, citrulline, right? Alanine, they all be able to generate it from glutamine metabolism in the intestine and release. So in other words, that intestine is able to release citrulline, alanine, right, and ammonium. And citrulline is released from intestine, basically goes to which tissue? Liver, kidney, brain, which tissue to get metabolized? Kidney, right? So this is the next slide. So the citrulline, so this is a partial, the urine cycle in intestine. So urine cycle intestine, because the two steps, the two enzymes that occur in the in the mitochondria, very active in the intestine, right? So the partial urine cycles, particularly first 2 steps, occur in the mitochondria that very active in the intestine. So we generate a lot of citrulline, right, in the intestine. And citrulline released into the circulation actually bypass the liver and directly go to the kidney for synthesis of arginine, right? So citrulline, again, synthesized in the intestine will not metabolize or pick up by the liver, but by a kidney, right, for synthesized arginine. And arginine then released back into circulation and pick up by other tissue for protein synthesis, right? Yeah, that's just the summary. Okay, so kidney. So how kidney metabolize amino acids? So kidney can also do the partial urine cycles, but they were not able to produce the urine, right? But kidney is a place that excretes the urine. Urea is synthesized in the liver and excreted in the kidney, right? But kidney also be able to excrete ammonium, right? So kidney also is an important place to get rid of ammonium, right? By release the urea, which is not synthesized in the kidney but in the liver, right? And also release or excrete ammonium. So ammonium is actually produced in produced in the kidney. So ammonium is NH4, right? And carbon metabolism. So carbon can be oxidized in the kidney, but also can undergo gluconeogenesis in the kidney, right? So these are the I think probably is important, slide. So 3 amino acids that can be synthesized in the kidney. Arginine is one of them, right? And then serine and histidine. And uptake of amino acid by kidney. So glutamine is mostly picked up by the kidney, and alanine, aspartate, the other citrulline, right? Glycine can all be able to pick up by kidney. And metabolic pathways, nitrogen, metabolism, I said, they undergo the partial urine cycles, and nitrogen lost primarily as a urine, right, under the normal condition. So, synthesized by the liver, but excreted in the kidney. And ammonium under the fasting condition, right, that become is also excreted, right? So carbon, just mentioned the oxidation and gluconeogenesis. So the slide for the metabolism in the kidney. So protein synthesis, right? The citrulline or aspartate, and also glycine, right? Uptake by the kidney and converted to or used for synthesis of arginine, right? So arginine is made from citrulline and aspartate, right? And then released to the circulation and goes to the other tissue for protein synthesis in the other tissues. And histidine can also be produced, and glycine can be converted to serine, right? Released. So this slide just tell us, the protein synthesis, what type of protein are synthesized in the kidney, and also the excretion of the ammonium, right? So ammonium is being released from, for example, from the amino acids like glycine, right? And then converted to ammonium and then released directly into the urine. And this is the slide showing the gluconeogenesis, right? It's also another different pathways, right, in the kidney. So glutamine is most important amino acids, right? Can be picked up by the kidney and converts to glutamate by glutaminase, and then involved in the deamination second reaction, right? Deamination by glutamine dehydrogenase. And the third reaction is the transamination. So it could go the opposite direction, right? And will produce oxaloacetate and the pyruvate. These are the two important compounds that can enter the gluconeogenic pathway, right? So you wanted to remember these steps, 1, 2, 3 steps, from the glutamine, and enzyme or reaction that involve these 3 steps, right, to convert glutamine to the intermediates of a gluconeogenesis pathway, right? So again, glutaminase and glutamate dehydrogenase and aminotransferase, right? So 3 steps to the deamination or transamination, right? And this is the partial urine cycles in the kidney, and basically this urine cycles, the reaction in urine cycles actually occur in the cytosol, right, in the kidney. It's different from the partial urine cycle in the intestine. So, and also I want you to remember the 3 enzymes, right, that involve the partial urine cycles in the kidney. So the first one is the arginine succinate synthetase, right? Basically in the urine cycle in the cytosol, right? And the second enzyme is arginine succinase, right? To break down arginine succinate into arginine and fumarate, and fumarate gets recycled, right, back to the oxaloacetate aspartate. And arginine, once it's synthesized, then released. So urine cycle in the kidney just stop at the arginine, right? Because the OTC enzyme is lacking in the kidney, right? So ornithine cannot be converted to citrulline. So ornithine is built up, and ornithine is the strong inhibitors of arginase, right? So the last step cannot be complete because of arginase activity is inhibited by ornithine, which is built up because of lacking, because of the lacking the OTC enzymes, right? So again, there's 3 reactions, the partial urine cycles, the 3 enzymes that involve the 3 reactions in partial urine cycles you wanted to remember, right? Okay. Yeah, so metabolism in the muscle. Basically, we're talking about branch chain amino acids, right? Because branch chain amino acids mostly metabolized in the skeletal muscle. So the Yeah, I think we already went through the branch chain amino acid metabolism in the muscle, right? So all the slide, that I described today, that's very important. I wanted to understand all of them. I will send you or post this review slides, right? So you can get the review slide. And again, try to focusing on the study guide and class notes. And if you have questions, just send me or Teresa's email, right? Try to answer your question as soon as possible, right?